Hello, Physiology 141 lab students. This is just a quick summary of what we're supposed to be doing for lab this week. First of all, I want to give a quick shout out to my morning students. I apologize for the problems created by the low bandwidth, but they have had that problem fixed here on the Windward campus, so we shouldn't be having that problem again. All right, so what do we need to do for this week? So we're working on week 4, cell and membrane transport. You need to watch each of these 4 modules that are on here. And the modules include an explanation of what diffusion is, what osmosis is, the properties that affect diffusion rate. And this first video will be the basis of next week's quiz. So make sure you watch that first video. You can define osmosis, define diffusion, and you can tell me the things that affect diffusion rate, like let's say molecular size, temperature, things like that. Also be able to classify tonicity, that is hypertonic, hypotonic, and isotonic. All right, so that's video number 1. Video number 2 shows you how to set up your agar gel. Hopefully you've already done that. You need to let that agar gel run for 24 hours. You need to take a picture at the very beginning when you've set it up, and then 24 hours later when you're reading and measuring those distances. And once you've taken those pictures, you want to go down to Dropboxes. And so Dropbox is located right here. And after you click on Dropbox, we want to go in there and say Actions, Upload Files. And basically, you're going to click on Drop to upload the file, and you're going to select your 2 Petri dish files, and we're going to upload those to Dropbox. So just click on the Petri dish, click Open. Now, here's the thing that people don't get. It doesn't actually upload until you hit Continue. Once you hit Continue, then it's fine. It's uploaded. You're good. So uploading the Petri dishes are half of your participation credit for this week. There's no participation quiz this week, but the participation credit comes from uploading pictures of your Petri dish, and the next thing comes from uploading your data. So if you go down to the bottom of the page, you need to go click on Upload Your Data here. And once you do that, it will take you to this web page where we enter your data for your blue number 1, yellow number 5, and beet extract. Now, if you see your name here, great, just enter it under your name. If you don't see your name, just go to the leftmost cell and then type in your name and then put in your data. And then once it does that, I'll make sure that all those data get incorporated into these graphs. I might have to go back and add some cells in there. But the point is, you need your data uploaded by Friday. And once those data are uploaded, we'll talk about them next Monday before we talk about what the lab report is going to be like. I'm going to give you 3 weeks from today on the lab report, so that should be plenty of time to work on that. Okay, so let's go back to lab. The only experiment that you're doing in person is that sort of 4.2 right there as you're doing the agar dye experiment. There are 2 more experiments, 2 involving osmosis, that I have filmed myself doing, and if you are doing this as a lab report, you do need to report on those. So you do need to collect the data from those 2 experiments, but it's involving watching me on the screen and then collecting the data as I read off the measurements for the scale, etc. So that's what you need to do. If you choose to do this as a lab report, you can then click on Guidelines for Osmosis and Diffusion Lab Report located at the bottom, and it will take you to this Word file that tells you exactly what should be in each section of that lab report. Because your lab report's worth 50 points, you only get 1 shot for it, so you want to make sure you do the best you can. The lab report itself will not be bullet pointed. If you want to see what the lab reports look like, look at the sample lab report, which was in our week 1 folder there. If you're having questions about where to find that, let me know. So just a summary of what you need to do this week. You need to watch each of the 4 modules there. You need to make sure you upload a picture of your gel at the beginning, at the end, and do that to Dropbox. And you need to input your data by 4 or 5 p.m. this Friday on the click here thing. After that, we'll talk about what goes in the lab report next week. And remember, what's on next week's quiz? Well, next week's quiz is based on video 4.1. So make sure you review that before you come in for next week's Socrative quiz. As always, if you have any questions or comments, send me an email or a text. My number is 808-429-6218. Talk to you soon.